I'm Emil Uffelmann from the Free University of Amsterdam, and I'm the lead analyst of the Alzheimer's Working Group of the PGC, and I'm happy to give you an overview of what our group has been up to recently. So last year we have published 2 papers where we mainly focused on the overlap of Alzheimer's disease with a number of neurological disorders as well as other forms of dementia. So Vera Forminik, she has looked at the genetic overlap of Alzheimer's disease with multiple sclerosis, while Doug Whiteman has looked at the overlap of Alzheimer's disease with ALS, Parkinson's disease, as well as Lewy body dementia. We have mainly been focused on collecting more data for the third freeze of our Alzheimer's GWAS, and compared to our latest publication in 2021, we have been able to more than double our effective sample size to approximately 370,000 at the time of this recording. We've also been focused on collecting more samples of non-European ancestry, and our sample currently consists of 10% of individuals of non-European ancestry. Now, this doubling of the effective sample size has allowed us to triple the number of loci that we discover. Now, within the next 1 to 2 weeks, we hope to finalize data collection, because at the time of this recording, we're still waiting for one more cohort that will add another 45,000 effective samples. So we expect that this will lead to quite a significant increase in additional loci. Once we receive that data, we will finalize and conduct our cross-ancestry meta-analysis, as well as a number of secondary analyses, such as stratified GWASs. Now, for the future, of course, we want to further increase the diversity of our samples, because at this point, again, it consists of 10% of individuals of non-European ancestry, but ideally we would like this percentage to increase together with the total numbers. We would also like to focus on more deep phenotyping strategies, so that we can contrast sort of these broad dementia phenotypes with more Alzheimer-specific ones. Now, finally, if you want to get involved in any way, please let us know. Thank you.
Hello, I'm Jack Hedema, here to represent the Anxiety Disorders Working Group. The most important thing we learned is that with sufficient time, effort, and patience, we could make it across the finish line. We posted our first meta-analysis on medRxiv in July, in which we found 58 genome-wide significant anxiety susceptibility loci in a sample with 122,000 European ancestry cases. This manuscript is currently under review at Nature Genetics. We have several important transitions occurring in our group. First, my co-chairs, Talia Ile and Jürgen Deckert, are stepping down. We are very grateful for their contributions that supported our progress over the past 7 years. I'm happy to welcome as new co-chairs Angelika Erhardt-Lehmann and Manuel Mathiasen. We are also happy to welcome 3 earlier career anxiety researchers to the group: Daniel Levy, Helga Ask, and Brittany Mitchell. Our core analytic group stays the same with Sylvia, Brad, and Nora, with the hope to expand our numbers going forward. We are addressing our diversity goals by beginning collaborations with PGC Africa, PGC Asia, and the Latin American Genomics Consortium. Finally, we plan to expand our anxiety genetics foci to include childhood anxiety, animal models, epigenetics, and neuroimaging. With our first genome-wide meta-analysis behind us, we will review secondary data analysis proposals at our expanded workgroup meetings this year. Next, we will be reaching out to our existing and new collaborators to assemble the PGC Anxiety 2 data freeze to begin to target individual anxiety disorders as well as dimensional anxiety, of course, conducting these analyses in cohorts of diverse ancestry. Finally, we hope to support all of these efforts with a renewal of our NIH R01 scheduled for review in the coming weeks. I want to put in a plug for our second major project on the genetics of dimensional anxiety measures used in clinical settings, led by Talia. We have thus far included data from almost 500,000 individuals and identified 47 genome-wide significant loci. Come hear more at the session on Thursday. Thank you for your attention.
Hi, my name is Barbara Franke, and here is an update from the PGC ADHD working group. We're almost ready to freeze the data for our new GWAS meta-analysis on ADHD, and this GWAS meta-analysis will approximately double the number of cases that we had included in the previous meta-analysis published in 2023. So we have about 75,000 cases with ADHD, and from this analysis, we can find a number of approximately 50 genome-wide significant loci. We also have data on from 23andMe to include in this analysis, and this brings our sample size up to 170,000 cases, and the approximate number of genome-wide significant findings also goes up to 170. Another study we're currently in the process of publishing is from the consortia IPSYCH and Impact within our working group, and it reports on approximately 9,000 individuals with ADHD. We find 3 genes associated with ADHD through rare coding variants, and those include MAP1A, ANO8, and ANC2, with substantial odds ratios, effect sizes. And if we put together the top findings from this study, we find enrichment of gene expression from these risk genes in GABAergic neurons, both immature neurons and mature neurons, as well as in dopaminergic neurons. The effects of the rare variants and the ADHD polygenic score are additive across all of the pentals of the polygenic score, as you can see in the left plot. That's it from this study, and we are working as part of the ADHD epigenetics working group, which is led by Tetiana Sayats, on meta-analysis for peripheral blood, cord blood, and saliva EWASs, and then we also want to combine those. We have data on somewhat more than 1,000 cases and 4,500 individuals from the general population. We are always looking for additional data, so if you have EWAS data, please come to talk to us. That's it for now. Thank you.
Hi, here's an update from the autism group. It has been a great year for autism research with contributions from many of our group members, and we have chosen a few highlights here. One is a nice paper by Patricia Kipkemoy from Elise Robinson's group on phenotypic and genetic analysis in the NeuroDev project. And Patricia will present on this at the Autism Symposium on Wednesday at 3 p.m. There's also a great paper by Jing Zhang on assortative mating, and 2 papers coming out of the IPSYCH group, one on polygenic risk of mental disorders and school grades, and another one on recurrent copy number variants. And then I'd also like to highlight these 2 papers. First one will be presented by Juan Jun at the oral session on neurodevelopmental disorders on Thursday, 1 p.m. And the very interesting paper by Barun will be presented at the poster session on Thursday at 5 p.m. So what about our core business in gene discovery? Well, thanks to a lineup of great analysts, we are hoping to have a final freeze very soon. We have currently a near final freeze that is almost doubling the number of cases and more than doubling the number of loci. We have currently 12 loci. We are currently incorporating additional samples from Spark, and you can hear much more about this at our Autism Symposium on Wednesday at 3 p.m. and also on our group meeting on Thursday at 11:30. Plans for the next year. Well, first and foremost, we want to finalize and submit our new GWAS paper, and then we'll follow up with a subsequent GWAS that will include ancestrally diverse samples, and this is led by Liana Hernandez and Mike Gandal. Obviously, we want to identify much more of the risk variation, including the interplay between common and rare variants, improve our understanding of the underlying pathophysiology, dissect the extreme heterogeneity of diagnostic subgroups, and dissect the genetic architecture of comorbidities especially. We also like to enhance the prediction of pertinent outcomes. So by this, thanks to everybody in the group, and see you there. Bye.
Here's an update from the bipolar disorder team. We finished the bipolar disorder 4 freeze, nearly 160,000 cases, 2.8 million controls, resulted in nearly 300 genetic loci, as illustrated here. Excellent job by a young analyst team led by Kevin O'Connell, also investigated the trans-ancestry samples. We also included analysis of different ascertainment types, from clinical ascertainment, in-house samples, community samples, medical records and health registries, and then finally self-reported samples. And it showed that the difference seems to be in genetic architecture, seems to be driven more by subtype. If it's bipolar 1 or 2, as illustrated here, than if it's clinically or community-based. Then we find some convergence between common and rare variants. And thanks to the great collaboration with the new functional genomics team of PGC, we identified specific enrichment of genetic signal in GABAergic interneurons and medium spiny neurons. And also an interesting link to enteroendocrine cells, which is something we will follow up on. Then the plans for next year, we will include more diverse samples. We are finishing the East Asia meta-analysis freeze now, have a meeting here at the World Congress, and also want to expand further the global participation. We have worked with the more deeply phenotype samples. We have a publication now for sub-phenotype analysis, very interesting work. We continue coordination with sequencing efforts and brain imaging, the Enigma Consortium, focusing more now on the longitudinal cohorts because of the intermittent phenotype of bipolar disorder. There we have a lot of potential for the biobank samples and especially birth cohorts. We are also a large team of method developers that focus on relevant use cases where we can develop some decision support tools. Thanks to all our participants in the bipolar team. Here, picture from last year. Please meet up at our working group meeting here at the World Congress. And please contact us if there are any questions or you want to participate. Thanks.
Greetings, this is Jonathan Sabat, Professor of Psychiatry at UC San Diego. I'm delighted to be able to share with you the results from our CMV analysis of 6 major psychiatric disorders. The intriguing results from these analyses show that all major psychiatric disorders have contributions from rare variants, both positive and negative associations for a variety of disorders. And typically, as is the case both for rare variants and common variants, a single risk variant is generally associated with multiple disorders. It's almost never specific to one disorder. When you distinguish between deletions and duplications, which is something that is particularly unique to CMV analysis, what you can see is a very interesting genotype-phenotype correlations. So where you have, for example, the CyFIP deletion that's associated with schizophrenia, you can see that that same deletion is negatively associated with bipolar disorder. So what you have is for one disorder's risk factor is actually negatively associated with another. And reciprocal duplication of SciFIP actually is positively associated with bipolar disorder and major depression. So these reciprocal deletions and duplications are really associating with different psychiatric spectra. And exactly the same is true for 22q, 16p, and a whole wide range of different risk factors. We're starting to be able to coalesce these things down into pathways using gene set-based analyses of cell types and brain region RNA-seq data. And what we can see is that the gene sets associated with duplication are, for example, frontal regions of the brain and temporal pole, and specific pathways like kinase signaling, histone modification, transcriptional regulation, membrane lipid, phospholipid metabolism, also interestingly, immune activation. Now, the deletion associations are a non-overlapping set. For example, in schizophrenia, at least, we're talking about visual cortex, synapse organization, calcium transport, neurotransmission. And of course, there are other pathways and cell types that are overlapping between deletion and duplication. But I think these very different brain regions and pathways that are associating with gain of function and loss of function may prove to be insightful for understanding how biological processes are affected in the developing brain and contributing to psychiatric risk. So, well, next steps, finish the 3 papers, incorporate trans ancestry analysis into all of them. And then meanwhile, the major depression and autism CNV analyses are being crafted into individual disorder studies. Thanks very much. And I look forward to seeing everybody at the meeting. Cheers.
Hello, everyone. I'm Jordan Smoller, and on behalf of my co-chair, Ken Kendler, I am pleased to tell you about the cross disorder group. The past year, the cross disorder group has mainly been focused on completing our extensive multi-method dissection of cross disorder relationships now among 14 psychiatric disorders, for which we had more than 1 million cases and about 5.5 million controls. And we applied a suite of statistical, genetic, and bioinformatic methods across levels of analysis, from the genome-wide level down to the regional level, to the locus level, and then functional genomic follow-up of those loci, to try to triangulate and get a clearer sense of the shared and more specific aspects of genetic risk for a broad range of childhood and adult onset psychiatric disorders. And there were, as you might imagine, a lot of very interesting results. I'll just mention a couple. We again find 5 latent genomic factors using structural equation modeling or genomic structural equation modeling that capture most of the variance across these disorders, and you can see them here. At the regional level, we saw many areas of the genome, regions of the genome that harbored shared genetic effects across disorders, including 101 hotspots, as we call them, that had significant genetic correlation among multiple disorders. One of those on chromosome 11 had significant relationships among 8 disorders. And in multivariate GWAS, we find 428 pleiotropic loci associated with these factors, as well as other loci that seem to be more disorder-specific, at least in pairwise analyses. And those were most commonly loading on different factors. And then in terms of the functional genomic follow-up, lots of very interesting results and findings. I'll just mention that we see some evidence for the hypothesis that the broadest level of genomic sharing is enriched for biological processes that are pretty general, like transcription regulation, but that as we get down to the factor-specific and maybe disorder-specific levels, we see more distinctive effects with different cell types that differ across the factors. So our next steps are to finally submit this completed manuscript. We're just awaiting acceptance of one of the other PGC workgroup papers. I want to give another shout out and special thanks to the incredible analysts who did an amazing amount of work on this. And the members of the cross disorder group, many of them came together to write another manuscript, a perspective piece on challenges and opportunities for cross-disorder research, which focuses a lot on some of the subtle methodological issues and lessons learned from this research. We hope to see that in the coming months. We are also discussing our next round of CDG analyses, hopefully incorporating things that we have not done before, like longitudinal trajectories, dimensional phenotypes, rare variants, and so on. And we're pleased to announce a leadership transition. Andrew Grotzinger, who really did a lot of the leadership on our CDG3 work, has agreed to be added as a co-chair of the group, and we may see further transitions as Andrew takes on that role. So that's it from the cross-disorder group. Looking forward to seeing everybody at this year's meeting. Thanks.
Hi everyone, I'm Laura Huckins. Together with Cindy Bulick and Zeynep Yilmaz, I'm delighted to co-chair the PGC-ED working group. Over the past year, our primary focus has been on finalizing Freeze 3 of our data. This includes our first analysis of binge eating behavior and a continued GWAS of anorexia nervosa. Both of these represent dramatic increases in case numbers, and we identify a number of genome-wide significant loci for both traits, including for binge eating broad, an association in the FTO locus, and for anorexia nervosa, an continued association on chromosome 3 with that really complex LD peak that we talk about a lot in our Freeze 2 paper. As a working group, our emphasis is still very much on expanding case numbers and increasing power. So we're excited to see a number of suggestive loci and hopeful that as our sample sizes increase in the next few years, we'll see those popping into significance. These substantial case numbers allow us to ask more interesting questions too, so we can compare between eating disorders and see how those architectures may differ and how they may be similar. So for example, we show that there are different genetic correlations between anorexia and binge eating across a range of psychiatric, substance, and personality traits. We see that associations with metabolism are unique to anorexia and don't seem to be significantly correlated with binge eating. We see different associations with sociodemographic factors between anorexia and binge eating behaviors. And we also see a number of significant associations with anthropometric traits in opposite directions. So anorexia nervosa is here on the left and negatively correlated, and binge eating behavior is on the right and broadly positively correlated with these traits. As a working group, we're especially interested to understand how BMI and how the genetic architecture of BMI interacts with the genetic architecture of eating disorders, and indeed how it may interact differently with the genetic architecture of different eating disorders. So in our upcoming manuscript, we use an approach called GWAS by subtraction to remove that BMI portion of risk and see how our genetic correlations change. So here I'm showing our genetic correlations for the full GWAS in blue, and in orange how those change or how they differ when we remove that BMI portion of risk. And you can see that broadly the anthropometric associations are removed once we remove BMI. We retain a few of those metabolism associations with anorexia after we remove the BMI portion, which is pretty interesting. And reassuringly, we see that most of our psychiatric associations remain the same after we remove that BMI portion. So all of these analyses, as well as analyses looking at genetic risk on chromosome X, looking at associations with subtypes, including restrictive and binge purge anorexia, binge narrow and binge broad, two different definitions of binge eating behaviors, and a number of other comparisons, including case-case GWAS and functional analyses, will all be included in our upcoming manuscript. Working group members should see that paper any day now. Hopefully it'll hit your email inbox, and we are really hopeful and excited to share that with the rest of you in the next few months. As I mentioned, our primary focus as a working group is still on expansion and on recruiting new samples. To that end, we've launched EDGI2, the Eating Disorder Genetics Initiative, that will recruit samples across all eating disorders across 10 countries. We also hope to launch LA EDGI, Latin American EDGI analysis that will recruit across Latin America. And we've launched a new working group led by Lisa Dinkler and Nadia Makali within PGC-ED to study ARFID specifically. Most importantly, though, we are excited to welcome new analysts, new trainees, anyone who wants to expand their work to think about eating disorders. Please do reach out either at the meeting or via email. We would love to talk to you. We would love to stay in touch. Thank you very much.
Hi, everyone. My name is Jens Henning Leffler.
And I'm Mary Ellen Linnell.
And together we lead the Functional Genomics work group. We started this year, and in our group, we take a very wide definition of functional genomics. Basically, we're interested in how to tie biology or mechanisms from the genetic findings. And we focus on what we can do together as a group or as multiple scientists instead of individuals, and also what we can achieve with long-term planning and a long-term perspective. So our goal is to provide tools and platforms for the PGC and the wider community to use. And right now we have 6 main projects. Me and Mary Ellen, we lead a project of trying to put together single cell data and putting that together. Yanitza Montalvo-Ortiz from Yale, she leads an effort to do meta-epigenome-wide association studies, mainly focusing on the methylome. Then we have John Mill from Exeter, who works on RNA and protein isoforms and long-read data. Haejung Won from UNC, which runs a working group on massively parallel reporter assays and trying to understand what they really mean. Then we have Shen and Lawrence, who leads a group on multi-layered QTLs, where they're basically trying to build a resource where we can have understanding of different types of QTLs from the same data. And Kristen Brennan from Yale, who leads the effort on coordinating IPS findings.
Now, there are lots of ways to get involved in the group. We are actively seeking new members who would like to be data analysts, data contributors, or speakers. So we begin all our monthly meetings with a 20-minute seminar speaker session. So if you're interested, please do email our research coordinator, Fatima Memich, and you can sign up for the different sub-work groups or to give a talk at the QR code on the left. Equally, if you don't have time to get involved, but you have data to contribute, on the right, you can see the QR code to our data census. So we want to see what kind of data sets are out there, where we have them in the numbers that it's really worth us bringing together and we can add something here. And in terms of this conference, all 3 of us are going to be here at Singapore, and we've got our group meeting set for Thursday. You can see the details there. And for a chance to meet us more informally, we will be at the Meet the Leads later that same day. And that evening, we're going to have functional genomics drinks at the lobby lounge at the Conrad Centennial from 6:30 p.m. onwards, so just after the conference ends. So that's a 5-minute walk from the conference center, and we're really looking forward to an opportunity to meet some new people, get together in person for the first time. So even if you don't know anyone in the group, do come along and say hi. If you want to know more about the group and what the individual work groups are doing, we did a worldwide lab a couple of months ago, so do take a look at the PGC website and have a listen. And yeah, take a photo of this slide and hope to see some of you at the meetings on Thursday. Thank you.
I'm Catherine Lewis from King's College London, and together with Andrew McIntosh from the University of Edinburgh, we have the pleasure of co-chairing the PGC MDD working group. A key focus for us this year has been finalising our multi-ancestry GWAS, more on the next slide. And in January, we published our large meta-analysis of diverse ancestry studies led by Carolina Kuchenbacher, which combined 90,000 depression cases from Asian, African and Hispanic groups, identifying novel loci, but also showing low transferability of loci from European studies. Jerry Gintovano published a GWAS on postpartum depression. Antidepressant response remains a focus with this study of CYP2C19 metabolizing status. And finally, we're always happy to support methodology papers where authors use the shared genotype data in the MDD group to test out new methods.
So this slide shows where we are with our current main GWAS analysis of depression in this PGC group. This is unpublished data, but you can find it as a preprint on medRxiv, and the title and Manhattan plot are shown on the right-hand side of the screen. So we have, in a trans-ancestry analysis involving all 5 major ancestry groups and mixed individuals, we've identified 697 independent SNPs at 635 loci, 293 of which are novel. This is a joint analysis of more than 685,000 cases of depression and more than 5 million participants in total. 23% of the participants are from diverse and mixed ancestries. The SNP heritability estimate using this data is 8.4%. And using polygenic risk scores trained in this sample, we can now explain more than 50% of the SNP heritability using polygenic scores. Our findings also identify and highlight potential repurposing opportunities, and we've been able to infer cell types by leveraging additional datasets from both mouse and human RNA sequence datasets.
For our next steps, we're starting to plan the accrual of studies for our next meta-analysis. Please get in touch if you have studies where you'd be happy to share genotype data or summary statistics. We want a full range of depression phenotyping from severe cases in clinical studies to much lighter phenotyped cohort studies. Projects already underway in the group include dissecting heterogeneity, looking at sex-specific genetic variants, treatment response, and molecular epidemiology work on a range of omics. We will continue our aim to ensure that the MDD group reflects the global burden of depression and includes all researchers worldwide, and will help people to get the training that they need within the group to be able to fully contribute to psychiatric genetics research. Finally, please do get involved. We're always delighted to work with new people and new teams. Thank you.
Hi everyone, it's time to hear about the PGC Outreach Committee, and I thought it would be nice for you to see some of the faces of who's involved in this committee. So I'm Helena Davis, bottom left. I'm the co-chair with Jerry, and this year we've been thinking a lot about our aims and our values as a team so that we can use these to guide us in the work that we've been doing. So our aims broadly are to improve the visibility, accessibility, and understanding of psychiatric genetics amongst both the general public and the wider scientific community. We also want to work to build trust in our community and to encourage involvement and participation in our research, particularly from underrepresented communities. Our values are inclusivity, integrity, transparency, collaboration, and communication. So for example, with inclusivity, we want to engage with and include a wide range of communities and recognize diverse perspectives, backgrounds, and experiences. And with transparency, we want to be really clear about the limitations of our research. So every year we organize and facilitate each PGC group, taking over the PGC Twitter, and this year we've tried to branch out a bit from this and have had groups like King's College London's Anti-Racism Working Group do a Twitter takeover, which you may have seen on Twitter. We've also organised our first ever WCPG symposium called Science Communication, the Importance of Language in a Diverse World. And this will be on Saturday, 9:45. So please do come along. We have some really, really cool talks lined up. And finally, we've created a new survey exploring the psychiatric genetics community's perspective on patient and public involvement in research. And you'll see the survey being advertised throughout WCPG. So please do take part, as we really want to hear from you. In terms of future plans, we want to really broaden our reach. So we'd like to, for example, connect with people outside of psychiatric genetics, but who are doing similar work in terms of outreach so that we can learn from them and not necessarily have to reinvent the wheel. We're also always keen for new members and ideas. So if you're listening to this and you think this work sounds up your street, then please do email me or Jerry or come and talk to us at the conference. We also want to better understand our community's perspective on outreach and organise some formal training in science communication. So just to plug our survey, once again, this is the slide that you'll see throughout the conference. Please do take part. It's about 10, 12 minutes long, and your opinion really matters. Thank you.
I'd like to thank you for the opportunity to present on behalf of the sequencing group. So the work we've done in the last year is a combination of method development and data analysis. So firstly, we have developed novel methods to incorporate pedigree information into designing into CNV analysis. which has been published. We're working on Bayesian methods of inference to improve analysis in pedigrees as well, and on pipelines for long-range sequencing in pedigree data. That's work by Niamh Ryan from my group. We have published data on MZ twins discordant for psychosis, identification of ultra-rare missense variants in US pedigrees. That's published. And using the Pekin pipeline, we've done a large-scale calling in pedigrees from a Mexican-American sample in collaboration with David Blan and John Blangero. We're working on other pedigrees, including this Costa Rican pedigree, and we've had some engagement with the PGC Africa group with regard to collaboration with them. I think what we know now is that it takes different methods to identify risk variants. We believe there's a role for large, multiply affected pedigrees, which may be particularly useful in identifying rare, biologically informative variants. I think we feel we've shown that there are better methods of pedigree analysis than just using straightforward case control methods, but the pedigree selection and phenotypes are important. So we're really interested in large-scale pedigrees where there's a mixture of phenotypes, including adult and childhood disorders, or incorporating developmental disorders or conditions such as epilepsy. We think they're going to provide the largest benefit in terms of discovery. And we are developing long-range sequencing methods to improve coverage. Finally, we'd like to work with more people. We would like to increase the diversity of the populations that we work with. We're working with some people in South America as well as with the African group. But the type of analysis we do, you can look at small pedigrees. It's not like you need the resources to sequence thousands of genomes to do this work. So we'd be interested in working with people, providing infrastructure and support if they want to do their own analyses. Finally, I'd say just come and see our symposium on the work that we and others have been doing at this meeting. And if you want to reach out, just contact me on the email detailed below. Thanks.
Hello everyone, I'm Caroline Nievergeld. I'm happy to present on behalf of the PGC PTSD working group. Our group has been very productive over the last year. First, we have published our third data freeze in Nature Genetics. Then we shifted our efforts towards receiving funding for the next round of projects. We were successful in renewing our parent grant for another 5 years. In addition, we received funding to expand the genetics of PTSD in African population. This effort in East Africa is led by Kerstin Köhnen and is also part of the Ancestral Populations Network. Next, to follow up on our findings, Nikos Daskalakis from the PGC PTSD Systems Biology Group is leading our next steps for PTSD genomics from loci to function project. And lastly, together with Jackie Meyers from the Substance Use Disorder Group, we are investigating genetic comorbidity of PTSD and SUD. We have also submitted a renewal application for our ongoing epigenetics working group led by Alicia Smith and a new project focused on TBI led by Seth Disner. PGC PTSD Data Freeze 3 is including over 1 million participants with over 150,000 PTSD cases. We identified a total of 95 unique genome-wide significant loci. These loci include genes related to neurotransmitter and ion channel synaptic plasticity modulators, developmental exon guidance and transcription factors, synaptic structure and function genes, and endocrine and immune regulators. Join us on Wednesday morning for our PTSD symposium if you want to hear more about it. Our plans for the year ahead. We are now ready to start the collection of data for PTSD Data Freeze 4. We will use these data to expand array-based GWAS and sequencing-based rare variant analysis. We will also continue to optimize the contribution of diverse ancestry groups, to perform functional molecular fine mapping, and finally to dissect the common genetic architecture of PTSD to reduce heterogeneity and strengthen polygenic risk scores. Thank you.
Hi everyone, I'm James Walters and it's my pleasure to be giving this update on behalf of the Schizophrenia Working Group with PGC. It's been a productive year. I've highlighted some of the outputs there on particularly important themes, and we're really exercised to think about this challenging problem of, particularly with diagnostic phenotypes, how they're validated in population and electronic health record data sets. And Sophie Legg's paper there has begun that process and shown that these can be considered in certain contexts valid diagnostic entry points. We're really keen for cross-group collaboration, and particularly with the CMV and suicide group. That's been productive this year and will continue to be. And then lastly, we want our data to be openly available, particularly to statistical methods development. And again, the Norwegian group, Hailang Huang's group, and Michael Zilla's group have all used the data very productively to develop cutting-edge methods that we also hope to apply to the emerging data sets. We've been asked to speak about what we know, and I think that's largely informed by the Nature 22 PGC3 study, together with the co-produced schema paper, and I think the schema partnership will only become more important in time. We've got an interim freeze plan to advance discovery further, and it's really pleasing that that is going to be close to 50% non-European samples once other samples are incorporated. And as well as that, the joint rare common variant discovery is going to continue to be important. Alex Charney's group produced the targeted sequencing paper last year, and we continue to work on that basis. We don't want to be constrained by some of the limitations of diagnosis, and so we're looking at stratified approaches based on treatment resistance as a definition within schizophrenia, but also by forming this new first episode psychosis group and advancing plans for the next stage bipolar schizophrenia analysis. And all this is underpinned by a greater understanding of functional genomics and the meanings of our associational signals, and so to support that work is going to prove to be key. We also want to continue to reach out and incorporate new collaborators and samples. The We Seek NIH grant, which is an affiliate grant for the PGC, and with Regeneron agreements now signed off, offers an opportunity for that. And there are also other grants that have spun out of the PGC, not least a recent treatment-resistant schizophrenia grant, and there are other grants in late submission stage. We're really keen, and I think we've effectively engaged a lot of early career researchers to access the data, but also join the activity of the group, and we want to continue doing that. And then lastly, our plans for the coming year. Well, a key deliverable for us will be that interim freeze. That's likely to include the addition of a new 40,000 cases, and that's without the biobank samples, so we really hope that may prove a key advance. We're moving towards greater availability of the phenotype data, and again, in time, that will become more important. We need to think about the methods of bringing in these various different data sets, and we've got a phenotype group considering that very judiciously with regard to diagnosis, and also doing the same on the analytic front, and deciding the pipelines and the sharing of code protocols to be able to have, as best we can, uniform analyses of groups that are coming in with some stat results is going to be key, and also the approaches to multi-ancestry analysis, which is thankfully now becoming a prerogative for us to be able to tackle. We want to continue supporting the groups, and particularly the continental leads, and we formed a group exec to promote that further. And the other thing is that if you want to learn more, come and meet me on Thursday, but our open meeting is a really good place to do that, which is being held this year on Wednesday at 11:30. Thanks and all the best.
On behalf of my co-chairs, Howard Edenberg and Joel Galanter, and our substance use disorders team, I am delighted to report that we had multiple publications led by our 3 NIDA-funded sites. We also want to take this opportunity to congratulate our early career investigators on their career development grants, the international awards that they received, and the faculty positions that they secured. So we've had a busy year. We've been working on a GWAS of DSM-diagnosed nicotine dependence or tobacco use disorder, for which we are refreshing with a new set of cases. Interestingly, what we found is that the genetic correlations between DSM nicotine dependence and other substance use disorders, which are typically diagnosed using ICD or DSM, were considerably greater than similar correlations estimated using measures of cigarettes per day or biochemical dependence, such as the Fagerstrom test for nicotine dependence. Using the DSM nicotine dependence measure substantially also improves the fit of our general addiction genetic liability risk factor. We're also going to look at the association between the GWAS and individual criteria for the DSM and the FTND using polygenic scores. And then this GWAS will be meta-analyzed with prior nicotine GWASs of quantity frequency, ICD-based tobacco use disorder, and the Fagerstrom. We are also part of a very exciting series of GWAS of stimulant and cocaine use disorder, which we eventually plan to incorporate into our general liability to addiction risk model. And finally, we recently launched sex-stratified analyses of substance use disorders. These analyses are in early stages of data curation and preliminary analyses, so stay tuned for those results next year. All of our GWAS have included data on diverse populations and genetic ancestries, and our recent nicotine analyses include both an African American and Asian GWAS component. We are also increasing numbers using All of Us, and we invested some of our PGC 3 carryover money to add diversity to our SUD cases. In addition, we look forward to working with the NeuroGrap researchers who are leading substance use GWASs in Africa. We're also eager to add substance use disorder data from multiple global biobanks. However, the unfortunate stigma associated with substance use disorders make them typically low priority targets for curation or genotyping in these international biobanks. We know, however, that substance use disorders are among the leading global contributors to morbidity and mortality, and we are certain that many of our colleagues here have potential comorbid SUD cases in their international cohorts. So we invite you to please join us tomorrow morning very early between 7 and 8 a.m. for the SUD networking meeting. Joel and I will also be around for the conference if you are not able to join that meeting. We also welcome your emails. We're really eager to brainstorm ways with you to expand the diversity in our substance use disorders data. Thank you so much, and we hope you enjoy WCPG in Singapore.
Hi, everyone. These are the updates from the Suicide Working Group. This last year, we published our first multi-ancestry GWAS meta-analysis of suicide attempt. And this had about 43,000 cases identifying 12 loci, and significant heritabilities both within and across ancestries, and also improved out-of-sample prediction using polygenic scores in non-European ancestry cohorts. These summary stats are available through the PGC DAC portal. And we also developed a phenotypic task group to start to more effectively define suicide phenotypes for use in genetic studies. So Eric Monson led this effort, which includes working group recommendations about the use of diverse types of information, like structured interviews, self-report, ICD codes, and so on. And he also led a validation of curated ICD codes with Vanderbilt and MVP. And this is now in review at Biological Psychiatry, and you can find that preprint on medRxiv. Now, with new funding from NIMH to Mount Sinai and other sites, we're now adding suicidal ideation to our analyses and also increasing our sample sizes for suicide attempt and suicide death, both increasing by about 50%. And the most recent freeze as of August has included new data from Brazil, India, and South Africa. And these meta-analyses are underway. Sarah will be talking about these this week at our symposium. She'll be discussing our meta-analyses, and you can hear more about what we're learning. And this session will also include on Wednesday updates from Chita Behera about postmortem suicide, blood, and brain tissue collection in India. And Gustavo Turecki and Hilary Kuhn will dive into functional omics and the use of electronic health records to better understand suicide mortality. So just prior to that session on Wednesday, please note that Melek, an analyst from PGC Africa, will be reporting on sex-specific GWAS analyses of suicide attempt in the PGC, Depression, Bipolar Disorder, and Schizophrenia Working Group data. And then we'll convene on Wednesday at 6 p.m. for our working group meeting. This is an open meeting, and you can join me and Neve on Friday as well for a meet and greet to learn more about the working group. So with the new funding and a larger group, we'll be focusing this year on publishing findings from the latest freeze. We'll be presenting on the usual follow-up analyses, looking at enrichment and function. We'll also be using GSAM and some other techniques to kind of disentangle the genetics of suicide from the genetics of psychiatric phenotypes. And Andre Shablin's going to be leading our sex-specific GWAS meta-analyses with help from Sarah and Melek and Atunu. We'll be building capacity in India with PI Chita Behera and his team. And Lucas Ito will be leading analyses of suicide CNVs, collaborating with the CNV group and leveraging data available from our group and from other PGC working groups. So please do join our calls. We welcome anyone who can join, and you can also email us to connect at the emails down here below. Happy to hear from anyone with interest in working together. Okay, thank you.
Hi, everyone. My name is Niro Kiwole. I hope you're having a great time in Singapore. On behalf of the Africa Working Group, I just want to give you a brief overview of some of our activities in the past year. In addition to broadening our membership base, we have in the past year increased our leadership committee. These are committees that are created with a dual purpose of interfacing with a larger global PGC community, but also with coordinating activities, research activities in specific disorders of interest and methods of interest in Africa within our membership. So we've increased our leadership in this area. We've also increased the number of our scientific advisory board. We are happy to announce that we have top class researchers, senior researchers across a range of research expertise in our scientific advisory board. Recently, we're happy to announce that we also have membership representation in the newly formed Functional Genomics Working Group, and we're already actively pursuing funding for collaborative projects. The past year also, our members have been involved in training activities. We are highlighting in particular this Welcome Connecting Science project, training project in June on polygenic risk score analysis, which our members took part in. This took place in Kampala, Uganda. It's been ongoing for a couple of years now, and our members have been actively involved in increasing the capacity, the expertise on polygenic risk scores in Africa. Of course, our members have been publishing. We provide here just a few of the really good papers that some of our members have published in the last year. There's a few more, but we just wanted to highlight this few. About it from us. Hope to meet one-on-one in Singapore and enjoy the rest of the conference.
Hello and welcome to the Cross-Population Analysis Working Group update. I'm Roseanne Peterson and I'll be giving this update on behalf of my co-chairs Hailang Wang and Lermy Duncan. So our group formed in 2018 and the membership has grown to over 100 plus members, and the goal of our group is to support a global network of researchers and to support genetic analyses across the ancestral spectrum by providing guidance and discussion on methods development, empirical investigations, and support of collaborative projects. So we meet monthly on the first Wednesday of the month via Zoom. Every third call is at the late time to promote global participation. Our meetings are open. If you're interested in joining us, please reach out to any of the co-chairs, and our emails are listed there at the bottom. So what have we been up to? We have created a network of over 100 researchers from around the globe. We hold monthly meetings, including educational talks. We've published a best practices paper and have engaged in outreach activities to promote diversity in genetics research and among researchers. Our monthly meetings are a mix of talks as well as project-driven, and here's a list of some of the most recent presentations. As you can see, these include educational talks on emerging methods, on data resources, as well as on ethical issues. And special thanks to our recent speakers, including Valentine Hebert, who presented on challenges in inferring between population genetic differences for complex traits from GWAS. Also to Dana Lepetu and Matter Singh, who held 2 sessions on the discussion of genomic data in the All of Us research program, including the infamous UMAP figure. Also thanks to Baran Gao, who presented on MeSusi, which is a multi-ancestry fine mapping method. One of our ongoing outreach activities is providing PGC website content on the importance of global population diversity in genetic research and among researchers. This is a collaboration with the PGC outreach group. We've created content for the general public and researchers. We have included frequently asked questions as well as listed resources, and a special thanks to the writing group for their contributions. Another outreach activity was participating in the PGC Train the Trainers workshop last year, where we provided an overview and tutorial on how to perform trans ancestry GWAS, exploring the why and mastering the how. This was co-developed by Raymond Walters and Matter Singh, and the recordings can be found on the PGC YouTube channel by searching for Train the Trainers. I'd like to close with highlighting some recent publications from our membership. We have extensive expertise in polygenic scoring. Example papers include PRS in Latin American and African ancestry samples, transferability of PRS across populations, and Mendelian randomization. Members also hold expertise in the genetic analysis of admixed ancestry individuals, as well as leveraging diverse genomes for fine mapping. We recently have posted about these key papers during the PGC Twitter takeover last month, so please check it out for the informational threads. That concludes our update. If you are interested in these topics, please join us. Again, our meetings are open. Our next meeting will be in person on Friday. Thank you, and we hope to see you at an upcoming meeting.
Hi, everyone. My name is Paula Justi Rodriguez. I'm one of the co-leaders of the Latin American Genomics Consortium. The LAGC was founded in 2019 to accelerate psychiatric genetics research in Latin America. Over these first 5 years, our network expanded from 25 to over 160 investigators, increasingly engaged in collaborative efforts. We have now secured access to over 400,000 samples from Latin American participants across US and Latin American-based cohorts and genotyped over 10,000 samples. We've also contributed to efforts to establish standard analytic pipelines for admixed samples. And recently, we completed our first meta-analysis of alcohol consumption on these 400,000 samples and have several upcoming meta-analyses across additional psychiatric traits and disorders. Likewise, we have several manuscripts underway, including an extensive review focused on the landscape of psychiatric genetics research across Latin American populations.
Hi, I'm Elizabeth Atkinson, co-leader of the LAGC Methods Working Group, and I'm excited to tell you about some of our recent efforts. So in the last year, we've been continuing compiling available resources geared at an analysis of admixed data that's reflective of the composition of Latin America. We hope to put that online and potentially publish a sort of short note for easy reference. Additionally, we've been building a streamlined computational pipeline for conducting ancestry-aware GWAS and Latin American data, with special phenotype considerations informed by each of our phenotype-focused subgroups. Again, this can serve as a resource for any groups that are hoping to run sort of tailored analyses like these in their cohorts. I've previously spoken at length about our efforts to ensure access to high-performance computing for all of our membership, and that's something we're still strongly supporting and working towards. Fresh off the presses, I'm delighted to announce that we have a recent preprint led by Jessica Maurer up on bioRxiv. So their work thoroughly evaluates features affecting local ancestry inference across many simulated demographic and data models to advise on best practice for conducting local ancestry inference in Latin American populations. Specifically, using simulated truth data sets, we tested multiple different demographic models. So looking at the number of generations since admixture, as well as changing up the admixture global proportions. Different features of genetic data, so for example, looking at arrays versus whole genome sequencing, as well as looking at the effect of imputation. And different reference panel compositions. So whether you have a small but very well matched reference panel versus a large but poorly matched one. And this was in order to quantify which factors most strongly influence local ancestry inference true positive rates, as well as advise again on best practice for implementation. We also characterize the most frequent error mode. So when you do get a wrong call, what's the direction? So importantly, we found that the Amerindigenous ancestry components, these green bars in the figure shown on the slide here, actually had a significantly lower true positive rate than other component ancestries. So just to sum up, improving local ancestry inference for admixed populations allows researchers to more accurately correct for population stratification in statistical models, look at demographic structure, as well as improve gene discovery, for example, with the tractor. And you can provide more sort of accurate phasing by incorporating this information. So with that, I'll thank you for listening.
Hi, my name is Janitza Montalvo Ortiz, an assistant professor at Yale School of Medicine, and I'll be discussing our progress so far. We recently submitted a review manuscript where we discuss psychiatry genomics in the diverse landscape of Latin American populations in terms of the prevalence of these different psychiatric disorders, as well as the different GWAS studies of psychiatric disorders and traits, including Latin American populations, where we see that the vast majority of these populations are based in the US. And we see a lack of representation of other countries, Latin American countries. And in terms of findings, we see that at least 3 studies that have been able to include close to 300,000 individuals and identifying genome-wide significant hits. We discuss gaps and challenges and propose potential recommendations and solutions to address them, where we go in more detail in the paper. So hopefully that will be out soon for you to read. We also are working very hard in our study on doing a large-scale GWAS meta-analysis of alcohol consumption, and this includes close to 500,000 Latin American individuals. It will be the largest to date. Last year we presented our analysis including 55,000 individuals, and now in just 1 year we were able to reach close to 500,000, where we see a significant improvement of the hits identified compared to previous studies. And we also discussed generic correlations with other substance use traits, and we see a varying transferability of polygenery scores based on different geographic subgroup of Latin American populations with unique and different ancestry admixture. So if you want to learn more about our efforts, these are the different poster sessions, science session, plenary session that we'll be participating at WCPG. We meet monthly, and if you want to learn more, you can reach our labs, our LAGC website here, and our Twitter account that can be followed. Thank you.